Not an affinity, I wouldn't say, but yes. So he moved to Edinburgh, progressing over the next 12 years to directorship of the MRC Centre for Regenerative Medicine, Edinburgh Neuroscience, the Wellcome Trust PhD programme in Translational Neuroscience, and the Dean of Research for the College of Medicine. He's an experienced grant panel chair, having previously chaired the committees of 4 major charities and the Wellcome Trust Molecular and Cellular Neuroscience Expert Review Group. His research is funded by Wellcome Senior Investigator Award and focuses on the biology of myelin formation in the brain with the aim of discovering novel therapies in that very important neurological disease, multiple sclerosis. He has trained many postdocs and students, and 13 of those students and postdocs now run their own labs in Europe or in the US. He was elected FMedSci in 2017, FRSE in 2018, and was awarded the Chancellor's Award for Research by Edinburgh in 2019. He moved in 2021 to become the Pro-Vice Chancellor for Medicine and Health Sciences at the University of East Anglia. A good move, I think. In 2022, he also took on the role, as I retired, of the Interim Dean in Medicine. And I would like you please to join me in welcoming Charles to give his lecture from smart wiring to multiple sclerosis.
Thank you very much, Bill, and thank you all for coming this evening. I'm very grateful for your interest and your support. So on my title slide, you can see the cell type that I've spent 40 years of my life, or nearly 40 years of my life, trying to study, the oligodendrocyte. I just think it's the most amazing cell in biology, and I hope by the end of this talk you will also be amazed by the importance of this particular cell type. So that is meant to work. Okay, so my research since I started my own lab has focused on these myelinated axons in the brain. So myelin you should think of as an insulation-like substance that is produced by cells called oligodendrocytes. And if you look at this panel on the left, you can see an oligodendrocyte schematically. And what happens is, at the end of each process, the oligodendrocyte elaborates this enormous sheet of membrane that wraps itself multiple times around the axon to provide what's called a myelin sheath. And these myelin sheaths are very extensive. About 50% of the frontal part of your brain is myelinated. And if you look at this digitized fMRI scan on the right, you can see the distribution of myelinated axons in the central nervous system. And what you can see very nicely is that these myelinated axons tend to be concentrated in the tracts that actually connect the different parts of the brain together. And because myelin is a very fat-rich substance and it's white in appearance, these tracts, when you cut the brain apart, they appear as white, hence the name white matter tracts. Or there is plenty of myelin in other regions of the brain, the grey matter, the cortex, etc., but it's not as extensive as it is in these white matter tracts. And for the purposes of our talk today, I'm going to be focusing very much on these areas of the brain. Now, the traditional view of myelin is that it has two major functions. One is to speed impulse conduction along the axon, and the other is to reduce the amount of energy that the axon uses when it's transmitting those impulses. And I'm very happy to talk about the physiology of that if anybody's interested afterwards. But what's become clear recently is there's a third, and I think absolutely fascinating part of myelin biology. Myelin, we think, is also involved in the brain's ability to learn, the most fundamental part, after all, the most fundamental property, after all, of the brain. And what we believe to be the case now is that myelination in the brain is set up with an initial pattern, which is hardwired, a bit like the design of your house. But then, when the brain starts using itself, when it's experiencing the outside world, those parts of the brain that are most active become more heavily myelinated than those regions of the brain that are less active. And in that way, those circuits that you actually use become more efficient, as a result of which the brain has learned. So it's not just synapses by which the brain learns. We believe it's also the oligodendrocytes and the myelin. And I think that means if you think of a computer where you've got all these wires rushing between the various boards of a computer, the brain is like that, but in addition, that wiring is smart. Hence the term smart wiring. If it's used more, it starts conducting better, faster, with less energy. So actually, I'll come back to this right at the end of my talk, but this is a really fascinating area of myelin biology. And those of you who heard me talk on Friday to the NIHA group, I talked about our work in that area at some length. But what I'm going to do tonight, to give you a flavour of my own personal journey in science, is I'm going to focus this talk almost entirely on multiple sclerosis. So I'm going to take you back to 1984, if I could. And two things happened to me in 1984. One of them is I got married, of which more later. The second is I met this gentleman called Ian Mcdonald. This is Professor Ian Mcdonald, who some of you may know. He's world famous for his work on multiple sclerosis. And when I was a young trainee neurologist at UCL, wanting to get more involved in research, I went to speak to Ian. And he very kindly took me under his wing. He was a really important mentor to me. And by taking me into his clinic and letting me see his patients, he introduced me to this fascinating but really potentially devastating disease of multiple sclerosis. And he showed me patients with MS, and you can see here what happens. This is the optic nerve of a patient with MS, with the myelin stained in silver, and you can see these large areas where the myelin has been destroyed by the inflammatory process that drives MS. He also showed me very clearly how the disease has two main phases. There's an early phase, which is relapsing, remitting. The disease comes and goes. But then gradually, with time, you start to see this secondary progressive phase of MS where you just get chronic increase in the amount of disability. And the time scale here is deliberately not shown because it can vary from patient to patient. The time to start progressive disease can be just a year or so, or it can be 30 years. And some patients are lucky enough never to enter it at all. But this is the basic pattern of MS, and the neurobiology of that I'll explain in a second, but in 1984 we didn't know it. We just knew what the clinical course was. So Ian was very kind to me and he let me work in his clinic, but I think he realized quite quickly that I wasn't actually a very good clinical neurologist and that really I was much more interested in the research side of it. So I remember one day he took me aside and suggested my time might be better spent if I went to do some basic research. And he suggested that I went to talk to this individual on the left. This is Martin Raff, who was a professor at UCL. He's a neuroimmunologist. He came out of immunology, and he pioneered the use of immunofluorescent labeling to identify neural cell types in culture. He was an absolutely pivotal figure in neuroscience for what he did. And so Ian suggested I went to see him. And so I did what you did in those days. I put on my best suit and tie. I packed my Samsonite briefcase, you remember those? And I went to see Martin. Now, Martin's lab was famously informal, and nobody wore suits or ties in Martin's lab. So they assumed I'd come to mend the photocopier. So I was shepherded off to the photocopier and told to get on with it. But after a while, the confusion was cleared up, and I managed to see Martin, and he sat me down in his office, and he started rabbiting on about these cells called oligodendrocytes. Now, I didn't actually know what an oligodendrocyte was at that stage, But I guessed it probably wasn't a very good career move to say that. So I took advantage of my medical training, which trains you to sound confident about things you know nothing about. And I managed to bluff my way, I think, through, because he offered me a PhD place provided I could get my own money, which in those days was a lot easier than it is now. And so Martin's lab taught me the glorious art of growing cells in culture, using this immunofluorescent labeling technique and taking photographs of them under a microscope using a 35mm camera and film. Again, something very few of you probably ever had to do. And very early on, I saw these two cell types here. This is a precursor cell, labeled with one antibody in red, and this is the earliest stages of an oligodendrocyte, labeled in green. And for me, this was love at first sight. I mean, once I saw these cells, I was completely hooked. It's absolutely amazing to be able to grow these cells in culture and watch them turn into this, which is a more mature oligodendrocyte. The nucleus is in red, and you can see all this myelin membrane in green, and the cell type produces huge amounts of myelin. I mean, this is nothing to what would happen in vivo. In vivo, if I put a beach ball in the middle of this lecture theatre, the oligodendrocyte would produce enough membrane to cover the whole of this lecture theatre easily. But in culture, they still do produce a lot of these flat sheets. Obviously, there's nothing for them to myelinate, so it's all flat. But the culture's really enabled us to do some really exciting things. So it was a wonderful PhD. And I think probably the most significant contribution I made in the 3 years I was in that lab was that I was able to show using tritiated thymidine to identify cells that were still dividing. I was able to show that actually in the adult central nervous system, we could see dividing precursor cells. So this is a picture of cells that I got out of an adult rat optic nerve, and you can see two cells there, both of which have got the autoradiographic silver grains of the tritiated thymidine causing silver grains to come out of the emulsion. Again, a technique that very few of you have probably ever had to use, because now we just use bromodeoxyuridine or one of its substitutes. But this was quite an important discovery, actually, because it told us for the first time that in the adult central nervous system, there were cells that could continue to produce new oligodendrocytes throughout life. And that's actually, as I will show you later on in this talk, an important observation. So by this stage now, 1986, I was completely hooked on developmental neuroscience and the techniques that Martin had shown me. So what I wanted to do next was I wanted to explore in more detail some of the molecular aspects of developmental neuroscience and see how whether or not there were any links between development and repair. So what I did is I went to Richard Heinz's lab at MIT, and we had 2 wonderful years in Boston, and Richard taught me basically molecular biology. And what I did in his lab is I was working on this extracellular matrix protein, fibronectins. Now, fibronectin is a key molecule in the soft tissues in our body. It's a classic extracellular matrix molecule, and it's made up of lots of different protein modules, each of which is coded by an exon. And as a result of alternative splicing of the RNA, there are a number of different forms of this fibronectin. There are at the embryonic form includes all of the exons, whereas the adult form skips out a number of the exons present in the mature molecule. And as a result of this, you create heterogeneity of the fibronectin types in the developing an adult organism. Now, I characterized that using in situ hybridization, which you can see in the top right here. But again, I think the most important observation I made was that I showed that if you actually injured an animal, you put a wound in the skin, which repairs itself quite well. I showed that there was a reappearance of the embryonic types of fibronectin once the repair process started to kick in. So I effectively showed that a repair was recapitulating development using the molecular biology techniques to look at the alternative splicing of fibronectin. And that got me really hooked on the idea of using developmental biology as a tool for studying regenerative neuroscience, as a tool for actually working out how we could repair the damaged central nervous system. And my enthusiasm for developmental neuroscience was further enhanced by a 2-week course I attended at Cold Spring Harbor in the United States. So the Cold Spring Harbor courses are famous for being really intense courses, and we had the most amazing 2 weeks of teaching from Corey Goodman, who's here on your left, and Paul Patterson, who's there on the right. 2 world-leading neuroscientists. And this is actually quite a fantastic group. Apart from this individual at the back here, that's me. Most of those people have gone on to be internationally leading neuroscientists. It really was an incredible group of people. I won't run through their names. I can remember most of them still. But the standard of that course was incredible, and I would recommend it to any graduate student or postdoc now. They are amazing courses. And this completely hooked me on developmental neuroscience. So when I went back to Cambridge and started my own lab in 1991, funded by the Wellcome Trust, that's what I wanted to do. I wanted to learn more about developmental neuroscience. And we focused on the extracellular matrix molecules that I'd become so familiar with in Richard Heinz's lab, and their receptors, which are a group of molecules called integrins. And over the next 10 to 14 years, we made a number of interesting discoveries about integrin biology in the central nervous system. I'm just going to show you one, which I obviously think is the most interesting, and that's what we found here. Via Barron, who was a postdoc in the lab, she showed that integrins amplify growth factor signaling. Now, that doesn't sound terribly interesting in its own right, but if you look at this dose-response curve, this is what she showed. So we were looking at a growth factor called platelet-derived growth factor, which is present in the central nervous system at a concentration of about between 0.1 and 1 nanogram per mil. Now, we knew from our cell culture experiments that those concentrations were not enough to elicit a response, in this case survival of the cell, if the cells were just grown in that growth factor concentration. The cells would still die. But if you at the same time activated integrin signaling pathways, you shift the dose-response curve by an order of magnitude to the left. And now what you can see is the entire dose-response curve lies within the physiological range of PDGF. So what that means is if the cells are growing on the right matrix, the growth factor signaling is amplified, and we showed how that was done, and that means that the cells are now able to sense and respond to that PDGF concentration. Okay, well, so what, I hear you ask? Well, what we actually showed was this is important for a really key property of the central nervous system. So we know that in the central nervous system, neurons and oligodendrocytes are massively overproduced during development. And the only cells that survive are the cells that wire up correctly or find their targets. There's massive death of most of the cells that are generated during development in the central nervous system. So the key question is, well, what are the mechanisms that are keeping these cells alive? Because we reasoned if we knew that, we might be able to get a handle on keeping cells alive in patients with MS. And it turns out that it's exactly that mechanism that I showed you in the previous slide. Because the oligodendrocyte, which you remember is going to myelinate an axon, is exposed to this very low concentration of the survival factors, which are not enough to keep them alive. But the surface of the axon is coated with extracellular matrix. So as soon as the incoming oligodendrocyte contacts the extracellular matrix on the axon, the growth factor signaling in that cell is amplified, it survives, and it can continue the myelination process. All the other oligodendrocytes that fail to contact axons die. And that provides a very simple mechanism for target-dependent survival, which is, as I say, a critical property of the developing central nervous system. And I think that was probably one of the most interesting things we did in Cambridge. But the problem was that after about 10 years of this, I started feeling, well, this is really quite interesting, but is it actually important? Thinking of Peter Medawar's famous dictum that any research is interesting, but very little research is important. And I found myself wondering, well, actually, is what I'm doing that important in terms of MS? And I was very struck by two other things that were going on in Cambridge at the same time. So firstly, the next mentor I'd like to introduce you to is Aleister Constant, who was the professor of neurology in Cambridge at the time. Very articulate, editor of Brain, a brilliant mind, was really establishing very clearly, along with others, what was underlying the neuropathology of MS. You remember I told you that it was relapsing, remitting at the beginning, and then you started to get this degenerative phase. Well, what he and others showed very nicely was that you can explain that very simply if you look at this diagram on the right. The normally myelinated axon is demyelinated by inflammatory cells in MS, and it's the inflammation that causes the relapses and remissions. It's not actually the loss of myelin on the axons that's causing the relapses and remissions. It's the activity of the inflammatory cells in the central nervous system. And once an axon has been demyelinated by the inflammatory process, one of two things will happen. Either in a natural regenerative process called remyelination, all of that myelin will be replaced and the axon will survive. And we know that happens in many patients with MS throughout their entire life. Or, because myelin is so important for the energy requirements of the axon, axons that are chronically demyelinated degenerate due to, at least in part, a failure of the ability to support their own metabolic functions. And it's that loss of the myelin and the degeneration that follows it that causes progressive disease. So MS can be thought of as a disease of two parts. There's the early part where the pathology is dominated by inflammation and the symptoms are caused by the inflammation. And there's a later part which is a straightforward neurodegenerative disease caused by loss of the myelin. And that neurodegeneration results from a failure of this regenerative process called remyelination. And that really made me think that I ought to be really focusing on remyelination and not studying development. And the other thing that made me think that is my friend and colleague Robin Franklin, along with his mentor Bill Blakemore, at the same time in Cambridge, were making really powerful use of a model that they developed whereby they could inject lysolecithin into a white matter area of an animal. And you get this very, very small area you can see here of demyelination. Now that area will heal very naturally and very quickly over the next 21 days. And you can see that in the bottom. You can see 3 light micrographs of the area. The one on the left shows the lesion before the toxin has been injected. And you can see those black rings, which are the myelin sheaths surrounding the axons. 3 days after you've injected the toxin, there is no myelin. The axons are still there. You may or may not be able to see that, but there is absolutely no myelin left in that lesion. But by 21 days after the injury, the myelin has come back. All of the axons are now remyelinated. Notice, if you would, that the myelin is thinner than it is before the lesion, and that is characteristic of remyelination. And again, I'll come back to that point right at the end. But this is a really beautiful model for looking at this natural regenerative process. And the really powerful thing about it, and what separates it from so many other models that we use in animals, is that the injury is instantaneous. It happens when you do the injection, and then it's over. There's no ongoing disease or injury process after that. Everything after that is regeneration. Whereas in many animal models, the injury process keeps on going. Some of the inflammatory problems, some of the dietary models, the injury process is very long-lived. And that makes it very hard to study regeneration because you've got damage and repair occurring at the same time. very difficult to separate those two apart. This model is great because you've got all of the damage at the beginning, and then all you've got afterwards is regeneration, and it's all happening in this beautifully synchronous phase. So what we did in collaboration with Robin's lab was we got involved in a project whereby you do the lesion, and then you basically section the brain at different times after the injury. And because you know it's so stereotyped, you can take a group of animals at 3 days, a group of animals at 7 days, a group of animals at 21 days. And then you use laser capture microscopy to cut out the area where the damage is, shown here in purple. And you can see the teardrop shape after you've cut it out. And you take all these little things you've cut out, and you sequence the RNA to see which genes are being expressed. And because it's also synchronous, you get beautifully consistent results. And then if you basically, what we did is we did something called a volcano plot. So what you're doing here is you're asking how much the genes change. This is comparing two different time points in this process. And at 0, no, the genes haven't changed at all. -5 means they've dropped, +5 means they've increased in expression. And then the y-axis is how significant are those changes. But that's very important. You want to know whether or not that change might have happened by chance. Obviously, if you've got a gene that's very highly expressed and it changes a lot, then it's very likely to be highly significant. So it's not surprising that the bigger the changes, the more likely they are to be significant. And that's why you get what's called this volcano plot, like lava spewing out of a volcano. And what I like about the volcano plots is it makes the bioinformatics so much easier because you just focus on the genes at the top right and the top left, the most significant genes that have changed the most. And now what we found was a large cluster of myelin genes shown in the circles here, and that wasn't a surprise because those genes we know will be expressed as the regenerative process kicks in. But the top hit, as it were, the top right-hand gene, was a completely new one called retinoid X receptor gamma, RXR gamma. And so what we did then next was to basically show that if you activate the RXR receptor in the oligodendrocytes during the repair process, you increase the amount of remyelination. And this work was done by Jeff Wang, who was a student in Robin Franklin's lab. We did all the cell culture work for this project. And you can see here, if you look at the lower photographs, you can see on the left, you've got the amount of remyelination that's occurred in the control animals. If you look on the right here, you can see the amount of remyelination that's occurred in the control animals. And Jeff has helpfully labeled in pink the axons that have remyelinated. And if you look following the injection of the activator of retinoid X receptor, you can see that there's a very big increase in the number of remyelinated axons, which was really very encouraging. And the whole thing is quantified on the right. So this was really very exciting because it provided a completely novel target by which we could enhance remyelination. And it also provided a potential route to the clinic because there was a drug out there called bexarotene that had already been licensed for human use, which was known to activate the RXR receptor. So we were very excited by that result. Now, as this work was going on, I had another lucky break, which was that Edinburgh basically got given £10 million by J.K. Rowling, Jo Rowling, and she was very keen that we established in Edinburgh a centre for multiple sclerosis research in recognition of her mother, Anne Rowling, who died of the disease. And she gave Edinburgh enough money to recruit somebody from outside to start this MS centre, and then a further gift to build a regenerative neurology clinic. And this money was used to recruit two people. I came first, And then about 1 year later, I managed to persuade a colleague from Cambridge, Siddharthan Chandran, I was one of the people who was involved in that persuasion, to come up as well. And it was a fantastic move for me. I was becoming quite frustrated in Cambridge by the lack of any opportunity for strategic leadership. And Edinburgh was great because it gave me that opportunity to start to build something, to start to build a centre for MS research. And obviously my focus was very much on the basic science side of this, but it was great fun. And one of the first things we did was we took advantage of the fact that in Edinburgh there were experts in an area of regenerative biology that we hadn't really explored well in Cambridge, and that was inflammation. I always think of regeneration as requiring three things. To understand it, you need to understand the stem cells that generate the new cells in the repair process. You need to understand the biology of the tissue that you're trying to repair. And you need to understand the inflammatory cells that accompany any injury and are a critical part of the repair process. So these three pillars of regeneration. And the inflammation was something we hadn't really looked at, but Edinburgh gave us the perfect opportunity to do that. And so a very talented postdoc, Veronica Miron, joined my lab. Veronica's now a professor in Toronto. And she had a background in neuroimmunology. And what she did was she asked a very simple question. We know that the microglia, which are the macrophages in the brain, come in two broad flavors. There's the classically activated macrophages or microglia, so-called M1s, that are pro-inflammatory. They actually produce inflammation. And I express a molecule called iNOS. And then there's the alternatively activated macrophages, which seem to actually dampen down inflammation. And they have a very separate set of molecules expressed, arginase 1 and the mannose receptor. And it had been known for some time that these two had very different properties, but nobody had really been able to tease out what was going on in the regenerative process because everybody was studying these molecules that mixed up damage and repair. And if you've got a mixed picture, then it's impossible to tell what these macrophages or microglia are doing. But of course, the model we had was perfect. And so what Veronica was able to show, as you can see here, was early on, you can see this in the top left-hand image, the microglia were all of the M1 phenotype, labeled in green, whereas by 10 days post-lesion, there was a switch to the anti-inflammatory M2 phenotype labeled in red, and you can see it quantified on the right. And then what she did was a really simple cell culture experiment that produced a dramatic result. She simply asked, if I take conditioned medium from the M1 and the M2 cells, and I put them onto oligodendrocyte precursors, what happens? And the result was really dramatic, and it's shown here. So this is the control on the left, and the top right is what happens if you put the M1 conditioned media on. Now, oligodendrocytes here are stained in red, and you can see that there's hardly any. But if you put the M2 conditioned media on, as you can see, you get this massive increase in oligodendrocyte generation. So that generates the very simple hypothesis that the switch from the M1 phenotype to the M2 phenotype that I showed you just earlier is critical for actually enabling the formation of new oligodendrocytes and accelerating the repair process. And if that hypothesis is right, then the prediction is that if we remove the M2s, we should slow down the repair process. And that's exactly what Veronica did. What she did, she used liposomes full of a very nasty toxin that kills macrophages, and she coated it with mannose, which binds to the mannose receptor that's only expressed on the M2 cells. So it's a magic bullet for killing those M2 macrophages. And what you find is shown in the image here. You can see that in the control experiment, basically you are getting good healing with the myelin marker, MAG, present at high levels throughout the lesion, at 15 days after the injury. Okay, so you get this nice green area where lots of myelin has been formed. Contrast that with what happens if you remove the M2s. You can see this area where the lesion is, where there is no myelin formation. So we can slow down regeneration if we remove these M2s, showing very nicely how important these inflammatory cells are for the repair process. So that was interesting, and Veronica's taken that work on to try to develop new targets based on manipulating the regenerative immune system in MS. But going back to Cambridge, we hadn't sort of given up completely. Remember that RXR study I showed you? So in collaboration with Cambridge, we were involved in a clinical trial of that drug bexarotene that I've just talked about, which activated the RXR receptor. This was a trial led by Aleister Constant in Cambridge. And what they found was quite interesting. Bexarotene did have a small beneficial effect on patients with MS. This is the conduction velocity in the optic nerve, measured by flashing a light at the front of the brain, flashing a light into the eye, and measuring when the impulse gets to the back of the brain. And the time it takes, obviously the shorter it is, the better conduction. And if you look at this graph here, you can see in the placebo group, there's very little change in time, but in the bexarotene group, there's a group of patients who've actually seemed to have responded quite well. The time has got a lot faster. But the majority of patients, it doesn't have any effect on. So what that suggested was that RXR gamma activation might enhance remyelination, but probably only in a subgroup of patients. And actually, I think this is a really nice bit of experimental medicine, because what we've done is we've discovered in the lab, we've discovered a pathway, we've tested it out in humans in a small clinical trial, and what we found is that the result was more complicated than we thought it would be. A small number of patients have responded, but the majority haven't. So that suggests that there might actually be some heterogeneity in those patient groups, such that some of them respond and some of them don't. So now we need to go and do more experiments to try to work that out. So when we do the next clinical trial, we'll separate those groups out. And to me, that's what experimental medicine should be. It's basically an iterative process where you go from the lab to the clinic, back to the lab, back to the clinic. And every time you do the loop, you get better and better results. So what we needed to do now was to investigate this question of patient heterogeneity. Now, that was not easy, because obviously you have to study human brain here to do this. And the conventional view of MS at that stage was that the rate-limiting step in the regenerative process, remember I told you that remyelination happens in patients with MS. The conventional view was that this was driven entirely by the differentiation of those precursor cells I discovered in 1984 when I was a PhD student. So the conventional view would be shown in the top here, that in an MS lesion, the precursor cells around the edge of the lesion become activated, they start to divide faster, they differentiate into oligodendrocytes, which produces new myelin, and the process of remyelination is complete. And people thought that that was the rate-limiting step. But we kind of already knew that probably wasn't as simple as that. My colleague Anna Williams, who was working in Edinburgh at the same time and has been a wonderful friend and colleague for many years now, did a very beautiful study where she counted the number of precursor cells in and around different types of lesions. And what she found, I think, is really important. She found this big variation in the number of precursor cells in and around the lesions. So the normal number of precursor cells you see in development in this graph is shown as the grey bar running across this graph. And here we've got different lesion types. You've got acute lesions, chronic lesions, and repaired lesions. And in all of the acute lesions and all of the repaired lesions, you can see that there are some lesions where there are more precursor cells than you would see in development, and others where there are less. But there's no consistent pattern. And I would argue that if this differentiation was the rate-limiting step, then you should see a consistent change in the number of precursor cells. You don't see that. So that suggests that the conventional view is oversimplistic. And it raised the next question we set out to look at, which is, well, where are the points of failure in remyelination? So what I've done in the top here is I've schematically represented a number of different stages of oligodendrocyte maturation. The first stage is the differentiation where they're actually formed, and then you get this incredible elaboration of their morphology. The first thing you see is this elaboration of processes, which was the wonderful thing I saw in cell culture in 1984, and then you get the formation of these myelin sheaths. Now, the hypothesis now becomes that you don't only see a failure in this initial differentiation stage in MS, but you also see failures in these subsequent stages in maturation. And the neuropathology already told us that that might be going on, because if you look in some lesions in the brains of patients who died with MS, and this is a slice of cortex, you can see the lesions in white matter by these holes in the blue stain used to label the myelin. You see these cells here on the right, where the oligodendrocytes have put out processes, they've run them along the axons, but they can't complete the final step. They're sort of stuck in this stage here in this schematic at the top. So, as I say, that suggests there are multiple points of failure. So we have to work out how to actually address where those points of failure are. Now that's difficult because we don't have antibodies that label each of these different oligodendrocyte stages. We have to find another way to do it. And so what we did is we used single nuclear RNA sequencing. So this is a technique where you take the brains of patients who've died with MS and you cut out, just as we did with the laser capture earlier on, you cut out the lesions, but now instead of sequencing the RNA in total, you separate all the nuclei in that sample and you sequence the RNA separately in every single one of those nuclei. An incredible bit of technology that is unbelievably powerful because it enables you to look at every single cell in that lesion and ask what sort of genes it's expressing. And this was work done by Sarah, who again now runs her own lab in Munich, in collaboration with Dihaj Malhotra from Roche, Gonzalo Castello-Branco from the Karolinska, and Anna, who I've already talked about. And the results were really interesting. In the first study, what we showed is that you could actually see quite clearly that the patterns of oligodendrocytes changed in the MS brain. So this is what's called a Tisney plot. And what it shows is the patterns of, it separates out the oligodendrocytes based on the similarity of gene expression. And you can use it to cluster cells. And what this is telling you is that there are 6 types of oligodendrocytes characterized by having similar patterns of gene expression. The oligodendrocyte precursors are down here, and there's an intervening cell type called a committed oligodendrocyte precursor. But what's interesting here is that those clusters change if you compare normal brains and brains from patients who've died with MS. And you can see that if you look at the oligo-1 cluster, you can see that it's very abundant in the control brains, but much less abundant in the MS brains. Conversely, oligo-5 has exactly the opposite pattern. Now that was important because it told us that we could look at oligodendrocyte heterogeneity in the brains of patients with MS. But it didn't actually answer the question in any way. It didn't tell us what the different, where the different points of failure might be, because the study simply wasn't big enough. So we're just completing another study, which I'm going to show you some of the results of now. And this is unpublished data now, although it is available on bioRxiv for those who want to see it. And what we did now is we studied a much, much larger set of patients. And critically, we also studied multiple lesions from the same patient to see whether or not we could identify any similarities within and between patients. And the results are, the preliminary results are very interesting. So now we've studied lots of different white matter samples. We've studied normal appearing white matter. That's the unaffected parts of the brain in patients who have MS, apparently unaffected, because we know they are affected. And of course, we've also studied control brains. And if you do that, you get, obviously, that I remember I showed you that plot of the oligodendrocyte clusters. You get a rather more dense plot because there are so many more cells involved now. You can see the different oligodendrocyte clusters here on the right of this balloon-shaped structure with the precursor cells on the left. And you can, using this technique, you can actually identify the sequence in which these different oligodendrocytes have been formed. You can form a maturation pathway, if you like, and we could show basically that you go from what three types we called oligo A, B, and C, where A are the first oligodendrocytes and C are the most mature oligodendrocytes. And then there's a type called oligo D, where the cells appeared highly stressed. But that didn't fit in the main pathway. That was off on one side. And now, if you take each individual lesion in the data set, and you can do this, of course, because you've got every single cell sequenced separately from every single patient, from every single lesion. Now you can start to ask the question, well, what's the pattern of the oligodendrocytes in the different lesions? And if you look at normal brain, what you find is that there are plenty of these mature oligodendrocytes in green, and then you've got varying numbers of the different earlier stages of oligodendrocyte maturation. But what's really interesting is when you look at lesions, now you find three distinct patterns. So in one patient, you might see a normal pattern, but in another patient, shown here in the middle, you get lots of these oligo Ds, those stressed oligodendrocytes. And in a third patient, what you find, this is the most interesting one in my view, you see a massive increase in those earlier stages of oligodendrocyte maturation, almost as though it's like a stream that has been dammed during the maturation process, and it doesn't go after that. And this is exactly what we would have predicted from the bexarotene clinical trial, where we saw a subgroup of patients who responded. That tells us that there's likely to be different groups of patients. As you can see here, because each of these clusters is one patient, in any one patient, the pattern in the lesions is the same. That's really important because it tells us in any one patient, if we can find the right drug for their type of MS, it'll treat all of the lesions. But it won't treat another patient who's got a different type of regenerative pathology, because there might be a completely different reason why the maturation process has failed. So what that tells us, I think, is there are three very important implications. The first is that what we were doing when we did the bexarotene study, one target fits all drug discovery approaches, that's clearly oversimplistic. And the problem with the regenerative therapy trials we're doing at the moment is we're lumping all these groups of patients together. And in we're doing that, we're massively decreasing their power because we're comparing the set of patients who might be predicted to respond to the drug with a much wider group of patients who we would not expect to respond. So we're unlikely to get a statistically significant result. What we need to do is we need to be able to separate out the different groups of patients before we actually start the trial, so that we focus only on those patients who we would predict from our studies in the lab would respond to that therapy. And for that, we need biomarkers for these different regenerative steps. So that's where we are with this project now. We need to identify biomarkers that can be used either from serum or from CSF, where we could start to split the patients into these three groups and then start to target the potential regenerative therapies. So, as I say, we're trying to get this work published at the moment. We're very excited about it because we think that we have actually discovered something really quite important, that MS is not from the chronic progressive phase of MS, which is due to a failure of regeneration, is not just one disease process, but there appear to be at least three disease processes. But in any one patient, you only got one of those three processes, which suggests that we will be able to target it. So that is very good news. So that's where we've got to. But where I've got to personally, obviously, is I've now been in this game 38 years, and I still enjoy it as much as I always did. And at heart, I'm still a cell biologist. I really want to understand the cell biology of these cells. So the next question I want to ask is this one here. I've talked about remyelination, and right at the beginning, you remember I talked about adaptive myelination. Now, one of the puzzling things about MS is the cognitive changes that you see in MS. So could it be, the hypothesis goes, that remyelin, this newly formed myelin, is unable to undergo this adaptive process that the myelin that was formed earlier in life is so able to do? And could this be one of the reasons why we see cognitive changes in patients with MS? And so that's a question that we're now looking at using various animal models to study whether remyelination, or remyelin as I call it, can undergo these adaptive changes in the same way that normally formed myelin can. And I think the results will be very interesting. So that brings me to the end of my journey, and thank you very much indeed for listening. But it wouldn't be appropriate for me not just to spend a few moments, Bill, I can see you're agitating, but I'll only be a few more moments. I would like to say thank you to a few people, because it's been a long journey and a very interesting one. So obviously I'd like to thank my mentors. For me, the most important bit of advice I would give an early career researcher, having learnt the hard way, is get a mentor and keep them really close. Make sure that they invest their emotional energy in your career. The mentor should not be somebody in your line management structure. It should be somebody who can look at you from a little distance and ask the question whether you're doing the right thing. I've been really lucky with my mentors. And when things have gone well, it's usually been because of my mentors. When things have gone badly, and they went very badly in Cambridge, I lost all my funding after 4 years, had to start my lab all over again. It was because I didn't listen to my mentors. I'd like to thank my past and present lab members. It's been such fun running a lab. We've had some amazing times. The things I enjoyed the most were our lab retreats, where we used to, because EasyJet and Ryanair were very cheap, and Edinburgh and Cambridge are both very easy, we used to go to really nice places. So this is my lab in Nice. We rented a villa in Nice for a week and had a wonderful week of art galleries and drinking and walking and a little bit of science. And it was these lab retreats were actually great. We took them quite seriously. We really tried to do some science, but we went to some wonderful places. We had also a fantastic retreat in Sicily as well. The Edinburgh neuroscience community was an incredible environment in which to work. I've mentioned some of my colleagues, but there are so many others. And you can see in this lower photo, you can see some of the myelin biologists at the Myelin Gordon conference earlier this year. So all these people are leaders of myelin research in Edinburgh. And it was an amazing group of people. You probably, Anna's there, you may be able to recognize her, but there are many others as well. And they're an incredible group of colleagues to work with. It was a wonderful mixture of collaboration, ambition, enthusiasm, and an over-fondness of Scottish malt whisky in many cases. But we had a fantastic time. But we were also, though I say it myself, we were quite good, and we really built up Edinburgh. So in the 2021 ref, Edinburgh Neuroscience beat Oxford and Cambridge in every single metric: size, quality, environment, the lot. I think that was a fantastic achievement, and a testament to an amazing group of individuals who it was a pleasure and a privilege to work with. I'd like to thank my funders. I was lucky enough to have almost continuous, notice the almost, I told you about the interregnum, continuous funding for 25 years from the Wellcome Trust, but I also had very significant funding from the MS Society, the MRC, the BBSRC, and the EU. I'd really like to thank the MS community. we worked so closely in Edinburgh with people with MS. They were key supporters of our Anne Rowling Regenerative Neurology Clinic. They put so much effort into making that clinic a success. And Jo Rowling herself really invested her time and her energy into making that clinic a success. So I'm hugely grateful. And lastly, I'd like to thank my family, my wife Jenny, who's here, and my 2 children, Mark and Sarah, who tell me they're listening, although I doubt it. And you know, they've put up with me and my enthusiasm for research for very many years, and I'm extremely grateful to them. And I'm also very grateful to you all, and thank you very much.
Now we have some time for questions, and there's a microphone going round. Please wait until you get the microphone before asking the question, because the people in YouTube can't hear what's going on until you speak into the microphone. So any questions, please, for Charles? Here we go, down the front here, please.
Thank you, Charles. Brilliant as always. Jane Preston, I'm a gynaecologist. I know nothing about MS, but you sound very sure that you might be able to find a biomarker within either serum or CSF. Why do you think you can get it with that rather than going to the biopsy?
The problem is that biopsying the brain, as you know, is not a trivial procedure.
Absolutely.
And I don't think it's something we can do as routine clinical management. So if I sounded sure that we would get a biomarker, I think I would probably want to rephrase it and say, if we're going to get a usable biomarker, it's going to have to be from one of
usable.
those 2 sources. I think even CSF could be problematic, because certainly in the UK, routine lumbar puncture in patients with MS is still not clinical practice, as it is in Germany or in the US.
Okay, thanks very much.
David?
Can I just follow that? How do you then look for those biomarkers if actually the data you've got that shows the way people behave differently is based on the pathology?
Yeah, so what we're making the assumption, aren't we, that there will be a link between the pathology and the expression of various proteins within the brain. So what we've done, I didn't show this, is we've looked at the genes that distinguish these 3 different patterns of the cellular composition that I showed you. And we've identified genes that seem to be expressed more in one of those 3 types than in the other 2 types. And now what we're doing is we're asking whether or not the proteins that are expressed by those genes are more abundant in that particular type of MF. If they are, then we go to start looking for blood and for serum. Okay? It's proving to be really difficult, and whether it's going to work, going back to Jane's question, I don't know, but it's the obvious place to start. We certainly managed to find genes that distinguish the different types.
So Charles, following up on that, you had 3 groups at the end there. Do you get 3 separate genetic profiles? Are they preordained in the way that they behave?
That's a very interesting question. So what we'd need to do to basically do that is we need to do whole genome sequencing of those patients, basically look for the genetic variants. The problem with the GWAS studies and the other studies of MS is that they've been focused very much on whether or not you get MS, rather than what happens when you've got MS, because we would predict that the 3 different types would have different genetic compositions, but the GWAS studies that have been done haven't been powered to look for that. They've just looked for the patients who were or were not getting MS. My prediction is that you're right, there will be a difference, but at the moment, we don't know the answer.
Any other questions? In the middle here? And then Max at the front. I'll give another 5 minutes academic leeway for questions.
Thank you very much for a really interesting talk. Gareth Matthews, Cardiology. Just thinking about the axon, the ion channel distribution is also heterogeneous. From memory, sodium channels more at the nodes, potassium channels more interspersed across the internodes. Does the oligodendrocyte and the myelination process drive the distribution of the ion channels in the axon? And does that have any differences for the physiological function of the nerve?
So that's a really interesting question. So I didn't talk about nodes of Remyelination because I didn't feel that I needed to for a talk on MS, but you're absolutely right. So the myelin sheath has a particular length, usually 100 microns or so, and the myelin sheaths are separated by a very small gap, about 1 micron, called the node of Remyelination. And exactly as you say, in that area where the axon is exposed, the voltage-dependent sodium channels are very highly concentrated, and the action potential sort of hops from node to node, which is why it's also energy efficient. The potassium channels are off to one side, as you say, and they're involved in the repolarization of the axon. So the question is, what role does the oligodendrocyte play in this incredible patterning of the axon? Now, the answer is not as simple as you might think, because there are unmyelinated axons in the brain which still have these clustering of these channels. So the oligodendrocytes aren't necessary for clustering. What we think they do is we think they basically sort out the spacing. The myelin sheath, if you like, almost by pushing the molecules, make sure that the spacing is correct. But there's a lot of work going on exactly the question you've asked as to how the oligodendrocyte causes that spacing. Because what I've just told you, there are some people who would argue very strongly, actually there's evidence that oligodendrocytes also do promote some clustering. So it's a controversial area. I'm sorry, I'm making the mistake of trying to be fair to everybody when I answer the question, and it's one of those areas where there are still lots of different views.
Sure. And another follow-up question, if I could. When you talked about the remyelination, you said that the myelin sheath was thinner in the remyelinated nerves. Does that have impacts on the space constant and the time constant, and is that what leads to the temperature dependency and failure at the internode?
It certainly is going to have an effect on the space and time constant, and we don't know nearly as much as we should do about the ephys of a remyelinated axon versus a normal axon. Whether or not that actually causes this phenomenon of temperature dependence, I don't know. Is it the inflammation? Or is it the properties of remyelin? It would seem to me that both could contribute.
Thank you.
Down the front here, Max, and then the gentleman in the red over here. And that'll be the last question.
Thank you. So fantastic showing the 3 different cell types that might be responsive to different therapies. Are there different types of therapies that line up with those 3 cell types, or are we not there yet?
Not there yet, no. And the only thing I would say is the type where you're seeing a very large number of what I'm calling stressed oligodendrocytes, you would have thought that some of the neuroprotective therapies that are being tried in some of the inflammatory diseases might actually be quite effective there, because there's obviously a huge amount of cellular stress in those oligodendrocytes. So drugs targeting cellular stress, I would predict, might be efficacious in that group, but in that group only. The other 2, the differentiation block, and if you like, what I call the sort of the trickle. One's a trickle and one's a dam, if you like. And I don't know at the moment whether or not, I don't know which drugs would be effective in which sort. We certainly have drugs that promote differentiation, but what effect they would have in the maturation block patients, we don't know at the present time. I think that's something we need to look at.
Last question.
Gareth Thomas, Orthopaedics. You've shown that demyelination is the result of inflammation. Big question, what causes the inflammation?
That is indeed a huge question, and a whole nother talk. I mean, we don't know what the trigger of inflammation in MS is. It is an autoimmune disease in that the T cells basically become responsive to the patient's own myelin. The most attractive hypothesis at the present time is that it's some form of molecular mimicry, such that one is exposed to a virus early in life, which epitopes of which are similar to the myelin epitopes in the brain, and then the patient becomes effectively responsive to their own myelin. But there's an awful lot that one has to explain for that hypothesis to be correct. Is there a virus? Well, there is some evidence. There was a recent study suggesting, looking at large numbers of American soldiers, there is some evidence that EB might play a role. Then there was, then you have to explain what's called the Latitude Effect, which is that MS gets more common the further away you go from the equator, and that's true both in the northern hemisphere and in the southern hemisphere. Then you have to explain the genetic effect, which is basically that if you have an identical twin with MS, then your lifetime risk of getting MS is about 30%. But if you just have a family member, a non-identical twin, a brother, sister, your lifetime risk is only about 1%. So there is a strong genetic predisposition, but we don't know what those genes are at this time, the GWAS studies I was talking about earlier. And the last thing you have to explain is if there is some sort of exposure that explains the latitude effect, it seems to be a childhood effect, because there is some evidence that if you grow up in a high-risk area and you move to a low-risk area as an adult, then you keep the high risk. But if you're born in a high-risk area and then early on in your life you move to a low-risk area, you have low risk. So it seems to be something that you're exposed to in the first 2 decades of life that determines that particular element of risk. So there's an awful lot we don't know about what causes that inflammation, but it's a really fascinating question.
Thank you, Charles. There will be drinks and nibbles outside, and we can continue discussion there.
Thank you very much.
I'm a bit worried that this slide is still up, Charles, because those members of my lab that are here tonight or online might think I'll be taking them to Nice or something like that for a nice wee jolly. I'm not sure that the budget's going to stretch to that these days. However,
well, the airfares have gone up a lot, but you probably couldn't afford it.
Yes. There's an old joke that I recount that says the best thing to come out of Edinburgh is the M8 going to Glasgow. It's the old Glasgow-Edinburgh rivalry. But I might have to reconsider that tonight, because Charles has really given us an exceptional lecture, and the work is clearly of the highest quality. So I would like you to join me and thank Charles for the inaugural lecture.